But yeah, so I'm going to tell you all about k-nearest neighbor graphs in high-dimensional data analysis. As a outline of the talk, I'm going to start off by introducing an example data set and using that to illustrate the curse of dimensionality, and talk about how k-nearest neighbor graphs help to overcome that. I'll then go a little bit into the mechanics of k-nearest neighbor construction and diffusion on the graph, which is important for Laurie and Yakir's method that they're going to talk about second. Then I'll finally illustrate applications of k-nearest neighbors to some practical applications. And finally, if we have time at the end, I'll go through how we approximate k-nearest neighbor graphs in practice, because our data sets are so large, brute force computation isn't really possible. Okay, so getting started here, I'm going to use MNIST as an example data set. And the reason it's a good example for people who are used to thinking about biological data is it's high dimensional, but it contains a lot of structure. In this case, clusters of digits. So each observation is a 28 by 28 dimensional image of handwritten text. It gets flattened out to a 784 dimensional vector. It's grayscale, so values range from 0 to 255. And like with single-cell RNA-seq data, there might be some clusters in here, there might be some continuous gradients, but they're hard to capture a priori. There's a lot of covariance in the pixels, but how do we represent them? Okay, so here is an example image. It's a 0, and here's the corresponding matrix of data values that would go with it. And this would get flattened out, like the rows would get stacked together into a vector. So now, what is the curse of dimensionality that we speak about? So the idea is that as the volume of space grows exponentially with the number of dimensions, that's way faster than the corresponding number of observations in our data set is going to grow. And so you're going to end up with a really sparse space. And when that happens, there are a bunch of really counterintuitive, paradoxical phenomena that can take place, such as the observations appearing all kind of similarly distant to each other in the high-dimensional space. So giving some practical instances of this, if you take a hypersphere and inscribe it within a hypercube, the ratio of the volumes of the hypersphere to the hypercube approaches 0. So if you start with a circle inside of a square, the ratio of their volumes is 0.785, and a sphere within a hypercube, it's 0.523. And as the number of dimensions goes up, the ratio approaches 0. And so this corresponds to all of the volume concentrating near the outside, the corners of the space, which is a very strange thing since we're used to living in a three-dimensional world. And one of the consequences of this is that the distribution of distances between points in the high-dimensional space concentrates in a narrow band. So, this is sampling points uniformly from a hypercube between 0 and 1 in 2 dimensions. You have a pretty good distribution of distances. But as the number of dimensions of the hypercube goes up, all of the pairwise distances between these uniformly sampled points concentrates in a very narrow band. So it's kind of an example of the paradoxical properties of distances in really high-dimensional space. And so illustrating that with the MNIST data, I'm showing here a few example digits, 0s and 3s, and the distances between them. And first you can notice that all the distances are between 2,000 and 3,000. And actually this 0 on the right is more similar to this 3 in the high-dimensional space than it is to the other 0. And so distance, Euclidean distance, is not capturing the similarities between the digits in a very simple, intuitive way. But those are just 4 example points. Here I've sampled 100 instances of each digit, and I'm showing the pairwise distances between them. And you can see that 0s are more similar to other 0s to some extent, but it's not all that impressive. And actually the similarities between, or the distance between 0s and each of the other digits is about the same. So distances is really not capturing our intuitive sense of which digits are similar to each other. And that's a consequence of the curse of dimensionality. Okay. So I'm just going to signpost here that the curse is going to cause problems for methods that rely on distance in the high-dimensional space. So that includes methods like clustering, such as k-means and Gaussian mixture models, and kernel density estimation. And so we're going to want an approach that kind of can help us to overcome that. And the key to the solution to the problem is recognizing that the vast majority of the data space is empty. So if we think about this data set, there are 70,000 observations and 255 to the 784th power possible data points. And the data points mostly look like this example on the left, which is like a randomly sampled point in the space, which has no connection to any number. So you could kind of think about the vast majority of the space having no bearing to the data set we're going to be looking at. So how does that help us out? So the step that we take as computational biologists is to make an assumption that the data set sits on a low-dimensional manifold in the high-dimensional space. And there's a lot of complicated topology in defining precisely the manifold, but intuitively you could think about it as a lower dimensional space within the high-dimensional space. So that might be arbitrarily warped and curved. So you could look at this example here of a plane that's kind of corkscrewing around in the three-dimensional space. It's easy to visualize a two-dimensional plane in a three-dimensional space, but in practice it's going to be something higher dimensional than that that's going to capture the possible variation of the numbers, of the digits. So you might imagine a space like smoothly transitioning from a 1 to a 9 and all the possible steps that could do that. And we could think about we're sampling data points from that higher dimensional space, like shown in the picture on the right. Okay. So a slight example of that is picking out this 0 I showed before and showing some of the other observations in ranked by Euclidean distance to the 0. And we can see that there's some sense of varying smoothly there. So these neighbors of the 0 that have a similar, that are minimal distance from it, have a similar kind of like lip in the 0 there. And that's why they are closest to it in the space. And similarly for the 3. Okay, so manifold is this complicated high-dimensional object, and we're not going to try to parameterize that exactly. What we do instead is use a graph as a discrete representation of the manifold. So on the left, we have points sampled from the manifold, and we connect points that are nearby each other in the high-dimensional space with edges to make a graph. And it's pretty apparent that that is able to capture some of the structure of this curving plane in the 3D space. And so when you flatten that out, it's preserved that topology very well. So in practice, we're going to use a graph as a way to represent the manifold rather than something more complicated. Okay, so that's kind of the overview of MNIST and the curse of dimensionality. And I'm going to come back to that throughout the talk. But let me first talk a bit about the mechanics of k-nearest neighbor construction. So here is an example data set. So a given data set will be observation by features, and each row is an observation. And I'm showing an example data set on the right that's only 2 dimensions. So it has an X and a Y feature, and I'm able to plot that in 2 dimensions really easily here. And so making the k-nearest neighbor graph is pretty straightforward. So for each observation xi, you identify the set of k other observations with a minimum distance to xi. So for this point here shown in red, I've just labeled the nearest neighbors of it, the points that have the minimal Euclidean distance to it, by drawing an edge between them. And it's easy enough to do this and to represent it in 2 dimensions. So we do that, in this case, k was 3, so I connected the 3 nearest neighbors. And you get a nice graph like this, where observations are nodes and edges connect the nearest neighbors. This is great for 2 dimensions, where you can see all the observations, but in higher dimensions, we need a better way to represent the graph. And we do that using an adjacency matrix, which is a matrix that's observations by observations in dimensions, and it has a 1 in the ij position if xi and xj are nearest neighbors, and otherwise it gets a 0. So here for our example data set, the first observation has a nearest neighbor in the second, fourth, and sixth position there. And so that's pretty much it for defining the nearest neighbor graph. It's pretty straightforward, but oftentimes, it's actually losing a lot of information about the data. So, by weighting each of the edges equivalently, you lose any information about which observations are closer to each other or further away from each other. So you might want something more like the graph on the right, which connects points that are further away from each other with a thinner line and points that are closer together with a thicker line. So that would be a weighted graph rather than an unweighted graph. Alex? Hey.
If you're going to go to as far as the one on the right, then why not threshold by distance instead of forcing everything to have 3 neighbors?
Why not threshold?
Like you're weighting by distance, and then beyond, but everything only gets to have 3 neighbors. But there's probably points that are closer that didn't get to be friends, and points that are further that did, because of the local density. I guess you're kind of accounting for the local density and making sure that those points don't get lost, or you can imagine different ways of doing it, I guess.
Yeah, it's a good question, and I guess you would end up maybe with some points that wouldn't have any connections at all, and then would be like off on their own. Yeah, I wonder if there's other formulations that do it that way, that just like calculate the distance between everything, and then set a discrete threshold. Yakir.
It does seem like there's I think this might be related to Alex's comment, I'm not sure, but there's like a tension here between the idea that initially we said distance is meaningless in high dimensional space, and now we're saying distance is meaningful, but like only very short distance is meaningful. And I wonder if that's somehow like, related to why it is that first you choose the 3, sorry, I should stay near the mic, like the 3 closest points, and then you like bring distance into the picture.
Maybe.
Yeah, well, one aspect of it is, I'm going to convert it to this weighted graph, and then we're going to talk about diffusion on it, which is going to allow you to reach further neighbors. And if you did that and didn't add some points that weren't connected to anything, you wouldn't be able to refine the graph through diffusion. So that'll make more sense in a few slides when I do the diffusion part. Okay, but yeah, really good question. But intuitively, we might want to preserve some of the information about the distances. And so how do we do that? We use a distance kernel function, which is essentially a function that takes a distance and converts it to a similarity or an affinity. So the most commonly used one is the Gaussian kernel, which I'm showing here, which essentially for 2 points that are identical, so if xi equal to xj, you would get a 1, and it decays exponentially from there towards 0. And how quickly it decays is determined by a parameter, sigma, which is a bandwidth that I'm not going to talk much about here, but it determines how quickly the distance decays, or how quickly the affinity decays with the distance. Okay, and so we can define an affinity matrix in a very similar way to the adjacency matrix. It's 0 everywhere on the graph except for nearest neighbors. And for nearest neighbors, you get this affinity function of the distance. And so for the example matrix I was showing before, it might look like this. Is this bar showing for everybody? Is that something I should try to hide somehow? It's good, okay, great. And this is a really important step towards our goal of a measure of locality on the manifold. So we were just saying that distance is not great in the high dimensional space, but we want a measure that says for 2 data points on this graph, like how similar are they to each other? How hard, and the way we're gonna frame that is, how easy is it to get from one node to another node on the graph? And that's going to circumvent the problem by being kind of an intrinsic version of distance that's on the manifold rather than in the high-dimensional space. And so the way we operationalize that is by asking, what is the probability of transitioning from node I to node J in a given number of diffusion steps? So how do we do that? We convert the affinity matrix to a Markov transition matrix. And so the equation for doing that is pretty simple. You start off with your adjacency matrix, which I showed before. You add to it the identity matrix, which allows self-transition, so going from node I back to node I. And then you normalize that, so each row sums to 1. So showing the example affinity matrix, you start off with that, but now you add to it the identity matrix, which adds 1s in the diagonal positions and doesn't change anything else. And then you normalize the rows to sum to 1, like so. And now the ij element tells you the probability of going from node I to node J in a single transition step. And that is kind of a measure of locality on the graph. How easy is it to move from one node to the other?
Why does the diagonal already have non-zeros there?
Yeah, depending on how you, so the question was, why does the diagonal by default in the affinity matrix have non-zeros, which amounts to saying, like, why isn't each node a nearest neighbor of itself by default? And I think different like software packages like manage that in different ways. But I think the default way I was thinking about it is you can't be the nearest neighbor of yourself. But I think certain packages like do have it included, but the ScanPy one doesn't, for example. Okay. So yeah, if you want to allow self-transitions, like allowing this node to transition to itself, you need to add the identity matrix.
So if I could just jump in for a second, I think maybe one possibility is like in practice, you're storing this as a sparse matrix. So why waste another like order n observations? You know, why like store another n ones in your sparse matrix when you could just not? Like that A matrix is stored in practice, like in a computer as a sparse matrix. Right. And if you stick ones on the diagonal, then maybe like if you're developing ScanPy, you just think of that as a waste of space.
Yeah, and also I think there's an intuition of whether a point is a nearest neighbor of itself or not. Like so. It's, but in practice, ScanPy doesn't include the self.
Yeah, I was just asking about the example on the slide.
Oh, like why Dylan chose to put zeros there?
Yeah. In any case, so you could think about that Markov transition matrix as specifying possible choices for a random walk. So starting from that example node, it can stay in place or it can move to any of its nearest neighbors, and the probability of moving to each of the nearest neighbors is encoded by the transition probability, like ij element in the matrix. And that's really cool. And it actually creates a really simple way of thinking about random walks of more than a single step. And that's going to lead us to diffusion on the graph. So it turns out that p squared corresponds to taking 2 steps on the graph. So multiplying the matrix multiplication of the transition matrix with itself is p squared. And what that corresponds to is what's the probability of moving from the start to some intermediate node times the probability of moving from that intermediate node to some other node j. And so if you look at what's the ijth element of p squared, it corresponds to summing over possible intermediate nodes x, the probability of transitioning from I to x times the probability of transitioning from x to j. So when you actually go through and compute that, you end up with this, these possible 2-step outcomes and the probabilities for them represented here. So why stop at 2 steps? You can now cube the matrix to get the 3-step transition probability. So the probability of getting somewhere in 2 steps times the probability of transitioning from that in 3, and all the way up to an arbitrary number of diffusion steps. But what does this kind of correspond to? Like, why might we want to do diffusion over a given number of steps? So this Markov transition matrix to the power t specifies the probability of diffusing from node I to j in t steps, which provides some measure of locality on the graph. like I mentioned before. And as t increases, the number of accessible points increases, and points that are further away in the high-dimensional space become accessible. So kind of the interpretation of that depends a little bit on what you're using the diffusion for. But one thing that we're frequently using it for, and what covariant neighborhood analysis uses it for, is for averaging signal on the graph. And in that context, larger t might decrease variance by averaging over signal from more points and points that are further away on the manifold. But it might increase bias because, like I said, it's averaging points that are further away. So that's kind of the trade-off you might be considering with the choice of t. Yeah.
So there's a choice in that self-similarity value, right? You can default it to 1, which, if you're in a sparse area of the graph, will increase self-transition. Right? Or you could do something that tries to always have the same probability, total probability of transitioning away, right? To sort of adjust for graph density or sampling density in that location, right? Assuming that your sampling process is sort of uniform across everything. Yeah. I don't know, has that been explored, like what your choices are for?
Yeah, well, I would just say that the choice of 1 is actually the value of the affinity of the Gaussian distance function of something with itself. So it kind of makes sense that it would be 1. But I think I understand the question. It's kind of like if you wanted to have faster diffusion, like having to diffuse away more, you might toggle that and use a distance function that wouldn't,
right, like if you assume that your sampling is sort of uniform in your possible space, then just because the points are more widely spread out in some location, you may want, like, adjusting that 1 allows you to sort of draw them back into almost a uniform density.
Yeah.
Am I thinking about it right?
Yeah, it kind of like it made me think about like why do we need to normalize the affinity matrix at all and convert it into a probability matrix? And I think it has something to do with density, like what you're saying, that in denser parts you would end up with like so much more diffusion like weights, and normalizing it corrects for that a little bit. But I guess you're right. If you can stay in the same place, you might end up doing that. But it's a really good question that I'm sure people have thought about. Yeah. Hey.
Hi. One other question.
Sure.
Why not using just the shortest path from 2 points?
Just the shortest path?
Yeah. Instead of computing, you're essentially computing some sort of. It's not even a shortest path. It looks like all pairs shortest path, but it's not.
Yeah, I mean, I guess you don't want it to be forced to only have one possible outcome. You kind of want there to be multiple possible transitions.
I mean the length. So if you just want to assess whether 2 points are close or far away, why don't you just compute it?
I think there are versions that like take geodesic distance, and it ends up being kind of a different variation on distance, but I think it also works.
I think geodesic distances are actually much more expensive to compute, and this integrating over random paths ends up being this beautifully simple linear algebra, which is part of the beauty of diffusion maps.
Yeah, that makes sense. I mean, so one thing didn't really get into, but one way of doing this really efficiently is to take the singular value decomposition, and then you can just be powering the eigenvalues in order to raise it to the x power. Yeah, in practice, because of sparse matrices, it's actually pretty fast to literally just take the powers of the matrices, and taking the SVD is kind of expensive too. So I was playing with both ways when I was like testing this, and I found it was pretty fast just to actually power the matrix directly because they're sparse. Yeah. In any case, so I think I kind of led to this that I'll show some examples of diffusion for different values of t. And I think the intuition for a choice of t is about decreasing variance versus increasing bias by incorporating points that are further away. So in practice, one of the main uses of diffusion is for averaging signal on the graph. So let's say we have some noisy signal v that goes with each observation xi. So I've colored each point here by this noisy signal vi. So multiplying matrix multiplication of the transition matrix V is going to smooth V by averaging it with points that are accessible in one step and weighted by the probabilities of those transitions. And multiplying P to the power T times V smooths it by averaging it with points that are accessible in T steps. So what happens with this data, you start off from the top, and it quickly smooths it, so you end up observing this signal that was noisy. It gets cleaned up, essentially. And I'll show this for MNIST in a second. But just to quickly summarize the mechanics of graph diffusion, you compute the nearest neighbor graph, you convert to an affinity graph with a kernel distance function, you normalize the rows to get a Markov transition probability matrix, and then you can calculate the probability of moving from node I to J in T steps by powering that transition matrix. And this gives you a measure of locality that's intrinsic to the graph, which is representing the manifold, as opposed to Euclidean distance in the high dimensional space, which is cursed. Okay, so back to the outline. I'm now going to illustrate application of k-nearest neighbors to a few common tasks that we want to do in single-cell data and in other high-dimensional data sets. So I just want to make the point that k-nearest neighbor algorithms are really widespread in single-cell data analysis. And I'm going to go over clustering and visualization at a very high level. And data denoising, and the main talk is going to be about kind of granular differential expression testing. So first, just to show diffusion on MNIST, essentially doing exactly what I just did for that toy data set, it can be used for denoising. So here's an example 6, which has some schmutz in it. Schmutz is a technical term. And with each round of diffusion, the schmutz gets smaller and smaller because the nearest neighbors on average are not going to have that same noise. So averaging with the nearest neighbors is going to end up cleaning that up. And here is another example. It's a 6 that has a lot of filled in part in the middle. And with diffusion, that gets cleaned up as well. But a thing to be wary of is if you diffuse too much, for certain cases, it could end up introducing bias. So here is like a strange looking 4, unfortunate looking 4, because it kind of looks like a 9, and it's going to end up having some nearest neighbors that are 9. And so when it's doing that smoothing based on the nearest neighbors, it's going to start to incorporate more and more 9s. And as you diffuse more times, you end up kind of closing off the top, and it becomes a 9. And so this is the same approach that is used for denoising single-cell RNA-seq data that was presented in a previous MIA talk by David Van Dyke and Smita Krishnaswamy. And here they are denoising single-cell RNA-seq data, which has a lot of 0 values in it. And the idea is by averaging cells that are nearby each other in high-dimensional space, or on the manifold, I should say, you end up revealing relationships between genes that are really hard to see when you look at the raw counts because there's so many zeros. And so you can see their talk for more detail about how they do that. Next, I wanted to talk about how k-nearest neighbor graphs are used to construct low-dimensional embeddings, such as TSNE and UMAP embeddings. And at a super high level, without getting into any of the details of these different methods, they both do something pretty similar, which is they take some high-dimensional distance or similarity function, which goes from 2 observations in the high-dimensional space and returns a distance, and a low-dimensional distance function that goes from, let's say, 2 dimensions, because we're often want to reduce the dimensionality down to 2 dimensions, and some cost function that wants the outputted distances to be similar to each other. So for TSNE, for example, that's going to be P times log P over Q. And then it tries to minimize the reconstruction of that. So it tries to find some values in the 2-dimensional plane of y1 to yn, that when you calculate the cost between points in their nearest neighbors, which is defined differently for the different methods. But it tries to essentially make those as similar as possible and solve some objective that tries to make the distances in two-dimensional space similar to the distances in high-dimensional space. Very hand-wavy, high-level version. They all have different choices, but they're trying to reconstruct the distances between primarily nearest neighbors. And this works really well for MNIST data in that in the reconstructed two-dimensional versions of these observations, it is able to preserve a lot of the structure that we expect to be there in high dimension. So for example, it keeps together data points that are from the same digit, and it reveals that there are these 10 different clusters of points in there. And beyond that, it shows some really interesting structure that might be harder to see otherwise. So looking at A, C, and E, like 3 points on the UMAP, they kind of live in this zone of confluence where there's 8s, 5s, and 3s that are all nearby each other. And looking at the digits that live in that kind of confluence zone, you can see why they're in a region where these different digits are nearby each other. It's because this 8, if you look at it, it isn't fully closed, which makes it look kind of like a 5. And the 5 looks a bit like a 3 that's had the top part of it chopped off. And the 3 looks a bit like a 5 where just this part was kind of a little bit too high. And so you can understand why they are being grouped closer together. And probably they have some nearest neighbors that are of the different digits. By contrast, B, D, and F, which are out in pretty far away from the confluence zone, are pretty clearly 8, 5, and 3 without that issue. Another part of the structure that gets preserved that I thought was pretty cool is if you look at the 1s, there's kind of a long, it almost looks like a 1, blob of the 1s here. And if you order some examples along that part of the UMAP, you can see that it's actually a gradient going from 1s tilted to the left to 1s tilted on the right. And it's able to preserve that structure really well. Okay, so this is some example of how 2D embeddings are able to capture that structure using the nearest neighbor graph. What about clustering? So I kind of signposted this before, but clustering that's based on the high dimensional distance is going to be suffering from the curse of dimensionality. So algorithms like k-means, Gaussian mixture models, those try to find cells that are close to centroids in the high dimensional space. But the conception of distance kind of breaks down in high dimensions, which can cause problems. Whereas community detection algorithms like the Leiden and Louvain algorithms instead try to cut the graph in smart ways to find regions that are highly interconnected but not as well connected to regions outside of them. And those are going to be intrinsic to the graph as opposed to relying on the high dimensional distance. And I didn't mess around with this too much to try to explore all ways to improve k-means. But you can see that, at least the Leiden algorithm worked pretty well for separating the points based on their true digit label, whereas k-means did not do as good of a job of that. And I can say for exploring a bunch of possible instances of k-means, it was not really happening for me. And you can imagine that might be because it's a malformed problem when you're using these distances that are, cursed. Okay, so that is 1 through 3, and I think we're doing okay on time probably. So I am going to give a little bit of intuition for how we approximate k-nearest neighbor graphs in practice. So when n is large, it's not really feasible to exactly compute all of the k-nearest neighbors. So you can imagine if we have 70,000 observations and we wanted to test each of them with each other one to see if they're nearest neighbors or not, that would be 4.9 billion comparisons, which quickly becomes infeasible. And we have single cell data sets of over 1 million cells now. we can't really look at pairwise distances as a way to find the nearest neighbors. So we have two main tricks that we do. The first is to run the nearest neighbor search on dimensionality reduced data. So for example, principal components. And that speeds up each computation because you don't need to do dot product of 784 dimensional vectors. You can do 200 dimensional or 100 dimensional vectors instead. But it doesn't fix that n squared problem. So in practice, we use algorithms that do approximate search in the space instead of trying all pairs. And the most commonly used algorithm is called approximate nearest neighbors. Oh yeah. And it is what's implemented in ScanPy and Seurat, for example, and it's able to reduce the complexity of this from n squared to n, which is really impressive. And these slides are all ripped directly from the originator of the Annoy algorithm, Eric Bernhardsen, who has a really nice tutorial on the method that goes into a lot more detail. But the intuition for it is, you don't want to be comparing points to points that are really far away, because there's no way they're going to be nearest neighbors, and that's the majority of them. So you're going to end up wasting a lot of time if you do that. The goal is to construct a tree data structure that will cause you to compare distances between points that are nearby to some degree. And we are willing to tolerate some inaccuracy if you end up with points that are not, that are actually nearest neighbors but aren't included. It's not the end of the world. So how does it do that? So the idea is to split the space into subspaces recursively. So it randomly selects 2 points and finds the hyperplane that separates them. And now the data is split up into 2 halves. And it does that recursively. So now it splits the first half and the second half, and so you end up with 4 quadrants. And at each step, it's storing the number of points that are in each part of the tree. And so you end up with a binary tree like this. And it repeatedly splits the space until there are at most k points in each leaf. And then it stops. And at that point, it has a binary tree that looks like this, that essentially has partitioned the space into regions that are likely to contain nearest neighbors, because the probability of separating 2 points that are close together is small. And at that point, you can find a query point in log n time by traversing the tree. So at each point, you ask, is my query point to the right or to the left of the hyperplane? Is it to the right or the left of the hyperplane? And you traverse all the way down the tree. And now you found the point in the partition with some candidate nearest neighbors. And in this case, there were 7 items, including the query itself, so 6 potential nearest neighbors in that same leaf, which is great if you are looking for 6 nearest neighbors. But what if you wanted more than 6? What it does in practice is it builds multiple trees at the same time. And with each one, it can search the query in log n time, which is really good. And so it ultimately takes the union of the possible nearest neighbors from the queries, the leaves that ended up with the query and across those different partitions. So it kind of looks like this. And now here are a bunch of candidate nearest neighbors, and it only computes Euclidean distance between those rather than between all of the points. And that gets you a pretty good speed up. It's possible that it's missing some of the nearest neighbors around there because of the way the data was being split up, but that's the trade-off for doing high dimensional, for the speed improvement. So definitely look at the tutorial for more details. It does some smart stuff as well, like moving down 2 branches at the same time if it's not getting enough nearest neighbors, etc. Okay, so let me just wrap up now with a few take-home points. We use k-nearest neighbor graphs as a representation of the data that's able to capture a low-dimensional manifold that the data lives in, and ignoring the truly vast empty space in a data set like MNIST. And we use diffusion on the graph to provide a measure of locality that's intrinsic to the data manifold rather than the ambient high-dimensional space. And this enables improved clustering, low-dimensional embedding, denoising, a whole variety of applications. And proximate nearest neighbors is useful for large data sets when we can't exactly compute the k-nearest neighbor graph. So thanks so much for listening.
Today, Yakir and I are going to be speaking about creating representations of single-cell data sets that allow us to better make comparisons across samples. And we first began this project with an observation, which was that people use a lot of adjectives to talk about single-cell data, that it's very high volume, that it's very high resolution, and also that it's very rich, suggesting it has some special kind of complexity. And we wondered what that term, richness, meant concretely about the structure of single-cell data sets. And that term could mean a lot of things, but we did recognize and then focus on one aspect of single-cell data sets that presents a particular challenge for their analysis. And to understand what that aspect is, I'm actually going to first start by talking about a more standard data set in which we've gathered a set of samples, and for each of those samples, we have a set of metrics that we have directly measured for those samples. So those metrics here become our features A through D. And if we want to compare our samples, we can look at these features. We can ask, for example, let's say half of our samples came from case patients, half came from control patients for some disease of interest. We can ask, are values of feature A higher on average in our case versus control samples? How about feature B? Or we can use these features in combination as input to some more sophisticated model. By contrast, in single-cell data sets, each sample is really its own data set, where our observations are cells, and what we've measured for each of these cells is expression level across genes, or something else, depending on what the modality is that you're using to profile your cells. The challenge that this presents is that there's no obvious way to collapse these data sets into a shared feature set across samples. We could, for example, because the feature set is actually shared across cells, meaning we're measuring across a fixed set of genes, we might be tempted to just collapse our cells axis and get something like the average expression for each gene in each sample, but that takes us right back to bulk expression data, so we don't want to do that. And we might also want to collapse that other axis, the genes axis, but we also can't do that immediately because there's no obvious equivalencies across our cells from the different data sets. So we need to do something else. And historically, the most common approach to this challenge has been to assign cells to broad categories, typically through clustering, and then compute the fractional abundance of cells from each sample in these groups. So for example, if this was a data set of immune cells, our groups might correspond to T cells, to B cells, to dendritic cells, and to monocytes. And research using this approach has yielded very important insights, but it does have a couple of also important limitations. The first being that clustering is done at the discretion of the researcher, who picks parameters like the clustering resolution that dictate the resulting cell groups. And another choice in the same immune data set that we're imagining might have led us to define a dozen different T cell subtypes and half a dozen different B cell subtypes. But the problem is that we don't know a priori which of these groups would capture the key difference between, say, our case samples and our control samples. In other words, there's no guarantee that clustering will capture the transcriptional structure that is most relevant for making our comparisons. The second limitation is that in doing clustering, researchers focus primarily on variation across cells, which doesn't necessarily correspond to variation across samples. You can imagine that one major axis of variation across cells could be stage in the cell cycle. But it's possible that in each one of our samples, we have approximately the same fraction of cells that are in each stage of the cell cycle. In other words, not only does clustering not guarantee that we will capture the cell states that are most relevant to, say, our case control contrast, it also doesn't even guarantee that any of our clusters will meaningfully vary across the total set of samples in our data set. So this inspires our driving question, which is, how can we compare samples in a single cell data set while making minimal assumptions about the transcriptional structure that's going to be most relevant for making those comparisons? Because if we can do that, if we can generate a set of shared features across samples, then we can plug this matrix into the array of existing tools that exist for analyzing something like that. And in particular, we might be interested in doing unsupervised analysis on this matrix to understand important structure and patterns of relationships across our samples. And we also might be interested in doing supervised analysis, like I've been highlighting. Maybe some of our samples came from case patients, some from controls. We want to understand what the systematic differences are between those sets of samples. So today, I'm going to start by introducing an emerging concept in single-cell data, which are transcriptional neighborhoods. And then I'll talk about how we define and use transcriptional neighborhoods in our method, covariant neighborhood analysis, or CNA. And then I'll pass the baton to Yakir, who will reveal and discuss what happens when we apply CNA to real data sets. So now that we've established the premise or inspiration for our method, I'll just pause quickly and invite any questions that you may have. And if there aren't any, we can just move on. Okay. So imagine that we have a single cell data set. In it, we have our samples. And for each sample, we have our cells by genes matrix. And just to build an intuition, let's imagine that we live in a world where cells only have expression across 2 genes. And because of that, we can actually visualize the generative distribution of cell abundance across our 2D gene space like this, where warmer colors reflect higher cell abundance. And then when we plot our observed cells on this gene space, they might look something like this in each of our samples. And our method is based on a key intuition, which is because the gene space is shared across these samples, that actually leads us to a very natural shared set of features across samples. Because even though there aren't direct equivalencies across any of our cells, what we can do is impose something like a grid on our gene space, compute the fraction of cells from each sample that fall into each grid square, and then those become our unbiased and shared set of features across samples. And of course, we may want to ignore all the areas of transcriptional space where we don't have actual cells by dropping those grid squares, or equivalently, we could actually sort of center a grid square on each cell in the data set. And that actually is helpful because it also lends us sort of proportional granularity to where we have greater density of cells in the data set. And you can also imagine that we don't necessarily have to use a square. We might use a circle or a Gaussian blob, but what we're trying to communicate here is that each of these regions becomes the area in which we compute the density of each sample. And what we're really doing is taking a kernel density estimate of the distribution that gave rise to the orange dot cells in each sample, where we're specifically estimating and storing the sample density that's centered at each of our observed cells. And each of these regions within which we compute the sample density, we call a neighborhood. And related ideas have been developed in parallel by the great folks that I've listed on the bottom of the screen here. And each of these neighborhood methods constructs neighborhoods and then uses neighborhoods in a slightly different way. So what do we do in CNA? Well, the first challenge that we contend with when we move from our 2D gene space to real gene space is that now we have thousands of genes, and it becomes impractical to actually define our grid and compute cell densities in that very high dimensional space. So as Dylan explained in his fantastic primer this morning, something that is really helpful to us here is a nearest neighbor graph, which allows us to retain information about the relative similarities among cells without having to actually keep the full gene dimensional information about those cell profiles. And we're trying to construct one neighborhood per cell in the data set. So let's just walk through how we would do that for a given cell. And I've highlighted one in red here that we'll define as the anchor cell for this neighborhood. And we want other cells in the data set to belong to this neighborhood, but we want their belonging not to be discrete, but quantitative, so that cells that are more similar to our anchor cell have a greater degree of membership in our neighborhood. And we can do this with diffusion. So as we're constructing our neighborhood for this anchor cell, let's imagine that we're also letting random walks start from any other cell in the graph. And we're interested in, for each of these other cells, it is able to take a step along any edge in the graph proportional to the similarity between, with probability proportional to the similarity between that starting cell and the next cell. In other words, sort of the weight of the edge. And so if we did this for all other cells in the data set, and we only allow those random walks to be a single step long, only those cells that are directly connected to our anchor cell are ever going to have any probability of arriving at our anchor cell. If then we allow two-step walks, then cells that are both directly connected or 2 steps away can gain non-zero membership in our neighborhood. But still, those cells that are directly connected and the ones that are most similar are going to have the highest probability of arriving at our anchor cell. So as we extend this outward, we get a probability density across all the cells in the graph that reflects the likelihood that a walk will arrive to our anchor cell from that cell after some number of steps. And we consider this to be the degree of membership for each other cell in the graph in the neighborhood for our anchor cell. And of course, picking k, the number of steps that we diffuse, is important to decide. And the way that we do that, I'll leave for the paper, I won't discuss in detail here, but essentially I'll share the sort of guiding notion, which is that we want our neighborhoods to be as small as possible so that we are modeling our space very granularly, but we need them to be large enough that we have some non-zero representation of most samples across most neighborhoods so that we can effectively make comparisons across samples.
Okay.
Yeah, please. Sure.
Oh, hi, I'm Brian. So you have weights in your graph. Mm-hmm. The edges are the k nearest neighbors. The weights are some similarity function, like what is that and how much does whatever you're doing downstream depend on that? Because I think that's like a big question. It's like, what's the right metric for how similar is cell A to cell B?
Yeah, exactly. So what we use is just the adjacency matrix from the nearest neighbor graph. So that's, and we use actually the default nearest neighbor construction from ScanPy. But you're right that that is sort of a key input to the method. So we sort of assume that you're making a nearest neighbor graph sort of to the best that you can given your data set.
So it's like Euclidean distance or something in 20,000 dimensional space, or what is the?
It's Dylan mentioned it in his primer.
Yes,
it's sort of this. That's it's like this affinity function. So like the Euclidean distance fed through a Gaussian kernel, I think, whatever the default there is, but I'm pretty sure that's what it is.
I see. Okay. Okay. Thank you.
Yeah. Thank you.
I think totally fair to point out that this is something that gets sort of like laid on to a nearest neighbor graph, and so the way you construct that nearest neighbor graph has to sort of like be thoughtful in the sense that it has to fit the properties of your data that you're trying to model.
Exactly, exactly. So now that we've talked about how we define neighborhoods using diffusion, let's watch it happen in a real single cell data set. And the exact specifications of what I'm showing you, the data set that's mapped here, don't matter. Well, they do matter, but Yakir's actually going to tell you about them later. But what matters for now is that we can watch diffusion happen to construct 6 neighborhoods, which we're starting with a single anchor cell for each. And then we can watch as we increase our steps in the graph, and we'll look at 8 steps here, that nearby cells start to gain membership in the neighborhood, but the cells that are further away have a lower degree of membership reflected by lighter colors in the graph. Okay, so we've talked about what transcriptional neighborhoods are and how we construct them in CNA. What I'll introduce next is what we do with those neighborhoods in CNA. So now that we've defined our neighborhoods, we can compute for each sample the fractional abundance of cells from that sample that fall in each of our neighborhoods. And this is our first representation, our preliminary representation of the single cell data set. where we have managed to capture greater granularity, more of this data set richness, and we do have a set of shared features across samples. But this can't be our final representation because it's still far too high dimensional. So we'll PCA it. And PCA is something that is going to be very familiar to many of you in this audience, but I'd like to talk about what it's giving us here in a concrete sense, because we find that PCA is capturing a type of information that we think is also biologically meaningful in a way that hasn't been captured in single-cell data sets before. So let's think about one of the two matrices in our decomposition here. For each of our principal components, we have the loadings of each neighborhood on that principal component. And those tell us about neighborhoods that are changing in abundance together across samples. So there'll be some samples where this group of neighborhoods are in high abundance together, say samples A and B, and then other samples where they're low abundance, or as we see in samples C and D, those first neighborhoods are totally empty. And you can imagine how our neighborhood loadings would be overlaid on our UMAP here, as I've shown. And we can imagine that some patterns of neighborhood covariance could look like this, where neighborhoods that are changing, that are positively correlated in abundance, are nearby in transcriptional space. That makes sense because cells that are transcriptionally similar are probably going to be sort of behaving in the same ways and responding to similar signals. But we can also imagine that there could be other patterns like this, where neighborhoods that change in abundance together are actually very transcriptionally distant. To take a biological example, let's say that some samples in our data set have higher signaling in some pathway, like the interferon pathway. And so what we see in that particular subset of samples is increased abundance in the cell populations that are responding to that interferon signaling. We see expansion of interferon responding T cells, interferon responding B cells, interferon responding dendritic cells, and those populations are all increasing in abundance together, but they're in very different spots in transcriptional space. Let's think about the other matrix in our decomposition, which are our sample loadings on these components. And our matrix in which we quantified the samples abundances across these neighborhoods, we call it the neighborhood abundance matrix, or NAM. So we call the sample loadings on these PCs, we call these our NAM PCs. And the sample loadings on each principal component tell us about the direction and the extent to which the corresponding pattern of cell state covariation appears in each sample. And this is our ultimate samples by features matrix, where our samples are featurized according to their abundances in each of the covariant neighborhood groups. And we see this representation as a key contribution of this work, because these PCs define the major axes of intersample variation in cell state abundances. And in this example that I've drawn, we've discovered that there's an important contrast in this data set between samples A and B on the one hand and samples C and D on the other. And this unsupervised analysis has revealed important relationships among our samples. I'd also like to highlight that in these features, these NAMPCs, we're still aggregating information about cell abundance across large swaths of transcriptional space, sort of like clustering. But the key distinction is the way in which we decide which parts of transcriptional space to aggregate together. Because in clustering, not only are those bounds set by the researcher, but they also focus on sort of contiguous parts of transcriptional space. Whereas our featurization is letting the data tell us which parts of transcriptional space, after we've subdivided it very granularly, we should aggregate together in our modeling, specifically because those parts of transcriptional space change in abundance together across samples. So these new features are therefore more likely to reflect meaningful variation across samples, and we care about that variation as unsupervised information about dataset structure, but we can also use it in supervised analysis. In a lot of these data sets that now are including single-cell data sets that are now including multiple samples, we also have some information about the the sources of those samples. Could be clinical status, like I've been referencing, disease case control status for some condition of interest. Could also be something, a continuous value, like a demographic value, what the age of the patient is from which those samples came. Or it could be an environmental feature, like whether or not the sample, the source of the sample was exposed to a treatment. And we can plug our existing samples by features matrix into any number of the tools that exist for analyzing that, for establishing or exploring these associations. And in CNA, we offer a built-in method to quantify associations between this representation, our NAM PCs, and any sample attribute a researcher may be interested in. And we do this through a relatively straightforward linear regression. So the real juice here is coming from the featurization that we've created, where we're regressing our sample attribute against the values, the sample loadings on each of our NAM PCs. And so from this model, we can get our effects per PC and a notion of significance. And it sounds simple, but and it is, but there are a lot of details here we had to work out. And I'll just signpost them, but we can save any discussion about what we actually did for either the question session or it's available in our paper. And some of those things that we had to work out were how to choose the number of top PCs to include in the model, how to deal with covariates to avoid confounding, how to guard against batch-driven associations, and how to define our p-values. But once we have that notion of overall significance, it tells us whether there is a relationship between something systematically changing in our single-cell data sets and our attribute of interest. But once we have a positive global association, we also want to know what are the cell states that are actually driving that association. And so to do that, we might want something like the correlation in abundance between abundance in each of our neighborhoods and our attribute of interest. But because we have so many neighborhoods, those become, and they're so granular, the actual correlations are going to be quite noisy. So what we do instead is actually first create a smoothed neighborhood abundance matrix where we just use the components from our decomposition that we included in our model. And then we compute the correlations between abundance per neighborhood in this smoothed NAM and the attribute of interest. And these become our neighborhood coefficients per neighborhood in the model. And again, there are some details to work out here. Namely, we use some linear algebra to compute these correlations efficiently, and we develop a notion of significance for each of these per neighborhood local associations. But overall, here is what CNA is offering to us. First, this unsupervised deliverable, a data-dependent and parsimonious representation of our single-cell samples, which are our covariant neighborhood groups as captured by the NAM PCs. The second is the opportunity to perform supervised analysis with this representation. And that analysis offers you both global and local association tests. To make that deliverable concrete, what I'm showing you here is a real single-cell data set. Again, you'll hear more about this one from Yakir in a second. But what you're seeing here is that in this case, there was a significant global association. And then when we look at the UMAP to look at our local associations, what we're seeing here is each dot corresponding to a cell, and each cell colored according to the correlation from its corresponding anchored neighborhood. And here, all of the cells are in gray, except those that passed our FDR correction. And those the neighborhoods that have a positive correlation to our attribute are shown in red. That reflects a population that has been expanded in our case samples relative to our control samples. And conversely, the neighborhoods shown in blue are those that have a negative abundance correlation to our attribute of interest, meaning that's a cell population that's depleted in our cases relative to our controls. So now that we've talked about what transcriptional neighborhoods are and how we make them in CNA, how we use them in CNA, and what CNA offers you, I will pass the baton to Yakir. Oh yes, please do.
This is Brian again. So a couple slides ago, you had like the regression on some features against the PCs and the betas, and I understood that you were doing some permutation tests there to get p-values on the betas here, presumably?
Yes.
But what you were just mentioning were p-values on individual cells. this is not on individuals. So what are those and how do you get those?
Yes, that's a great question. I think we're going to save exactly how you're right that we do that separately. They're actually also empirical p-values based on permutation testing that's done in a slightly different way, and it's done separately for our global association and for our local associations, which are done on a per neighborhood basis. And we're happy to talk about that more in the discussion section at the end.
Okay. Thanks.
Yeah.
All right. Thank you, Laurie. I'm now going to talk a bit about how CNA performs on real data now that Laurie sort of described it to you. And in doing so, I will be skipping over some of the simulations that we have in the paper, looking at null and non-null situations to kind of validate the method, just sort of for the sake of time. So there are 3 data sets that I'll be showing you analyses of today. The first is a data set of fibroblasts that are sampled from joints of patients with rheumatoid arthritis, so that's an inflammatory condition causing joint pain, versus patients with osteoarthritis, so non-inflammatory, or at least that's how we think of it, joint pain. The second is a data set of peripheral blood mononuclear cells from patients with and without sepsis, which for any folks who might not know what that is, they can think of it as sort of an extreme state which an infection causes runaway inflammation that damages the body. And the last data set is a set of memory T cells collected in a pretty large set of patients who are infected with tuberculosis. And I'm going to focus on different aspects of the analysis of these data sets in kind of a progression. So in the first data set, I'll be focusing on sort of an unsupervised analysis using CNA. In the second data set, I'll focus more on a supervised case control type analysis in which we use CNA to identify cell populations that are associated with the sepsis phenotype. And then in the third data set, since it's larger and extensively phenotyped, I'll actually show results from both an unsupervised and a supervised analysis. So let's start with the first data set. It consists of about 27,000 fibroblasts, and they're profiled from the joints of 6 patients with rheumatoid arthritis and 6 patients with osteoarthritis. And in the published analysis of this data set, the authors used a very sophisticated trajectory analysis with a lot of domain expertise and parameter tuning to eventually determine that these fibroblasts actually lie on a transcriptional spectrum. And over on the right, what you're seeing here is a UMAP of the data set with cells colored basically according to where they lie on the spectrum. Now, using some experimental approaches, the authors eventually showed that the spectrum actually seems to correspond to the degree of activation of these fibroblasts by notch signaling proteins that are produced by the blood vessels in the joint. And down in the bottom, I'm showing you here the same UMAP, but colored by an experimentally derived score of notch activation. And you can see that the trajectory that was found by the authors corresponds decently well to this ground truth experimental score with a correlation of 0.43. Now, I won't get into the details, but I'll say that this notch activation gradient turns out to actually be highly relevant to the rheumatoid arthritis phenotype, and it spurred a lot of follow-up work into the pathophysiology of this disease related to the interplay between the vessels in the synovium, the joint capsule, and the fibroblasts that lie in that capsule. Our question, of course, when we looked at this data set was basically whether this sort of notch activation gradient would somehow show up somewhere in our NAM-PCs. And sure enough, it actually turned out that NAM-PC1 seemed to capture the signal pretty well, actually even a little bit better than the original trajectory-based analysis with a correlation of 0.56 to this experimental ground truth. So in this case, we kind of achieved the same result as the trajectory-based approach, but without any parameter tuning. This was with CNA default parameters. And in addition to finding this spectrum, CNA also was able to prioritize it for us because it shows up as PC1, and PC1 was actually quantified by the PCA and CNA to explain 39% of the variance in the NAM, so 39% of the intersample variation in the data set. So this really, CNA is telling us that this is a dominant axis of variation in the data. And as a comparison point, we also just applied standard PCA to the cells by genes matrix that you would get if you just smushed together all of the samples. And PC1 there had some correspondence to this notch activation gradient, but it was worse with a correlation of 0.22. And we think this demonstrates concretely the difference between looking at transcriptional variation across cells, which can include sources of variation like the cell cycle that Laurie kind of gave as an example, which are the same across samples, and explicitly looking for variation across samples the way CNA does. So This is kind of the first analysis, just because it's the first real data thing. I'll pause to make sure this all kind of made sense and take any questions. Yeah, Dylan.
So I'm wondering, was there a reason to think that the notch activation signal should vary across samples? Like, is that an RA versus OA thing, or is it something else, genetic, or like why do you guys think the notch signal varies across samples?
Yeah. Okay, so I think first, I guess I should caveat my answer to say that obviously, like, it's not like the only things that are biologically interesting are the things that vary across samples. Like, those two are a Venn diagram. But I think that in this case, probably because it turns out to be related to RA, and the data set was ascertained in a way that it has, 6 RA patients and 6 non-RA patients, it's sort of, you're kind of, because of the case control design, like the things that are going to vary across samples will probably end up being the things that are relevant to RA versus OA. And so I think that's kind of what's happened here.
So you found it in an unsupervised way, but it reflects the case control.
I think so. And I think more broadly, maybe like I think this is responsive to what you're saying. I think what you're making me want to emphasize is, like, of course we keep giving the cell cycle as an example of like variation, intercellular variation that's not intersample variation, but it's not, that sounds dismissive, and it's maybe a little bit like oversimplistic, because obviously, like, yeah, there's probably lots of, gradients reflecting really interesting fibroblast biology here that are not varying across these samples. Yeah. Okay, any other questions for the moment?
Sorry, the correlation is specifically what?
So this is good. This is the kind of question I want. I want to make sure we're on the same page. So these correlations are basically across all the cells in the data set. Oh, sorry, Brian asked, what are the correlations? What are these correlations that I put up on here? And so just to take an example here, this R equals 0.43 is the correlation across all the cells in this UMAP, basically between the coloring here and the coloring down here, which is the kind of like experimental thing that we're considering to be the ground truth. Does that make sense? And then this is 0.56 is correlation between this sort of labeling of the cells and the ground truth, and then here 0.22 is this labeling of cells and this ground truth. So it's basically us just trying to quantify what I think is visually apparent, which is sort of that, there's different degrees of correspondence here between the kind of like inferred cell labels and the ground truth.
And does notch signaling show up if you just run this on either cases or controls separately? Does notch signaling or notch expression?
We did not do that. That's a good question. I wonder.
Yeah, I think I would expect that it would show up. I think my question would be sort of what order would it be in the major components of the matrix? And I think what we're able to emphasize here is that when our data set includes the cases and controls and therefore emphasizes the case control contests, it pops out as NAMPC1.
Yeah, that's a cool idea. Okay, let's press on. So I'll now talk about the next data set, our second data set, which we analyzed with a lot of help from Joyce Kang, another member in the Raychaudhuri lab who's pictured here and who I'd like to acknowledge. And in this data set, we have about 100,000 PBMCs from 65 patients, about half of whom had sepsis and about half of whom did not. And I do want to say this is a really lethal phenotype. Think, patients in the ICU with failing organs. And so when this data set was put together, actually by folks at the Broad, it was really an unprecedented resource for discovering and describing the molecular correlates of this condition. In the published analysis of this data set, there were a lot of comparisons across many different subtypes of sepsis, each of which had its own results. But the dominant theme among these was enrichment of a specific monocyte cluster that I've shown at the bottom here, the MS1 cluster, among patients with sepsis. And now I'm showing you the results of the CNA-based analysis of the data. It's clear, of course, that CNA just visually also finds enrichment of this MS1 group of cells. But what's really interesting about these results to us is that CNA also finds a lot of heterogeneity within many of these authors' clusters, many of these author-defined clusters. So let's see what I mean. Here, I'm showing you the same CNA results 4 times, but in each case, I'm graying out all the cells except for the ones in the indicated cluster. So for example, on the left here, you're seeing the CNA results colored just for the cells in the MS1 cluster. Then over here, you're seeing the CNA results just for the cells in the MS4 cluster. Here, you're seeing them just for the cells in the B cell cluster BS1, and here you're seeing them just for the cells in this T cell cluster TS2. And what we see is that though for MS1, there is really a uniform trend towards expansion across the cells in the cluster in sepsis, the other clusters don't behave this way. So in MS4, we have a subset of cells that's expanded, that's these red cells, and a different subset that's depleted. And in BS1, we have, and in TS2, we have a subset of cells that's more strongly depleted than the cluster at large. So there's kind of this white area, which are cells in the cluster that don't seem to have a very strong local abundance correlation to the sepsis phenotype. And there's another way, let me just show this in one more way that's a little more quantitative. What I'm showing you here are histograms of the neighborhood level correlations for the cells within each cluster. So on the x-axis of each of these histograms are the correlations between neighborhood abundance and the sepsis phenotype, and the y-axis is frequency across cells in the relevant cluster. So here we've got the histogram just for the cells in the MS1 cluster. And again, and the vertical bars, I should say, are sort of the FDR significance thresholds. And so you can see that in MS1, again, there's a broad trend towards enrichment across the whole cluster in sepsis. But over in these other three clusters, there's a really interesting, I think, bimodality. And there's sort of a clear trend towards different degrees and even directions of effect in different subsets of these clusters. So what we're seeing here is kind of a depiction of cases in which maybe these clusters were not the ideal way to partition transcriptional space in order to understand relationships to the sepsis phenotype. And overall, 8 of the 15 published cluster-based cell states that we looked at included subpopulations with these kinds of distinct abundance associations to sepsis. And I do want to mention that this clustering was not like a throwaway, we'll just pick Leiden resolution equals 0.5 clustering. This is a clustering that was really created by experts in this field with even different clustering resolutions for different cell types. So I think monocytes and T cells, for example, were given different clustering resolutions. So this was a very thoughtfully put together clustering. So overall, I guess, stepping back, I think what we like about this analysis is that here CNA was able to both bypass the sort of parameter tuning that I just described and to provide results that are pretty granular. And each of these identified populations could, of course, contain biology that helps us understand why patients with sepsis do so poorly. Are there any questions about this analysis? Okay. So I'd now like to move to our third data set. It's a larger, more extensively phenotyped data set, and it consists of about 500,000 memory T cells collected from a Peruvian patient cohort. And these cells were profiled with CiteSeq, which, as a lot of you probably know, simultaneously quantifies both mRNA transcripts and also a limited set of manually chosen cell surface protein abundances. And in the sort of main analysis conducted of this data set, these two modalities were merged using CCA. Now, these cells were collected from just over 250 patients, all of whom were exposed to TB, but only some of whom, the progressors, went on to progress to bad disease. And this was kind of the primary phenotype that folks who collected this data set were interested in. But the data set also includes other demographic information like age, sex, and a few other things that we'll talk about in a minute. So given the size of this data set and its relatively not unprecedented, but sort of a new degree of focus just on memory T cells, we were really interested, first of all, to just ask what is PC1 of this data set. And here's a UMAP of the neighborhood loadings of that PC. And this PC actually turns out to explain 24% of the variance in the data set. So in a data set of this size, that's actually a 65x enrichment over what you would expect if the spectrum were sort of trivial. And when we looked at this and sort of talked to folks in the lab about it, we noticed that the positive loadings of this PC, the neighborhoods that have positive loadings, they tend to be in UMAP regions that correspond to cytotoxic T cells. So the T cells that actually kill the other cells, the business end of the immune system, like the first authors of the T cell world. And then these blue neighborhood loadings tend to be kind of in the T helper cell part of UMAP space. So those are the cells that manage other T cells, the PIs. And when we saw this, that's actually this is a distinction obviously that's been around for a long time. And in recent years, it's actually kind of turned out to, many folks have started thinking about it as more of a spectrum. And so what we decided to do was kind of try to compare NAMPC1 to one of these scores that scores T cells along this kind of PI to first author gradient. It's called the innateness gradient. And we found that this PC actually correlates really strongly with that measure of innateness. So the correlation here is 0.81 again across all the cells in the data set. And to us, this was really cool because innateness has been discussed a lot as a major axis of variation across T cells, but our result actually shows that different people differ considerably in the average degree of innateness of their T cells. And this is a new result that suggests that this type of variation across T cells is not only important to our understanding of how they work, kind of on a biological level, but also perhaps to our understanding of why different people's immune systems differ from one another. Another neat thing about this PC was that we found that it was strikingly consistent across the three modalities in the data set, so the mRNA, the protein, and the CCA. So what you're seeing here is a plot in which each dot is a sample. Yeah.
This is Brian again. To go back to the last slide.
Yeah.
So you mentioned that this analysis shows something about the degree of innateness in different individuals'
Yeah.
T cell populations. I mean, you have this score of innateness that's derived from something.
Mm-hmm.
If you just this fact that the degree of innateness varies from
Across
individual
samples.
to individual, did the NAMPC sort of uniquely identify that, or if you just apply whatever you used here for the innateness score without
Yeah,
the
for
NAMPC?
sure. Yeah. Like you're saying, if you just computed the average, if you took a bunch of this data set and computed manually the average innateness score across the cells in each sample, you would find the same thing. That's absolutely true.
Right. Okay. That's what
Yeah, I think what we're saying here is not that this is the unique way to like prove that, but rather that this led us to that sort of finding.
Sure.
And I guess in a way also quantifies that it's like, the 24% quantifies that it's really a dominant axis of variation in the data set. But if you were, that's sort of like a hypothesis generating claim. If you were just wanting to test the hypothesis, you wouldn't need any of this fancy stuff.
Right. Okay. Thanks.
So let me come back to this plot for a minute. This is us demonstrating that this PC actually is sort of basically the same across the three modalities in the data set, the two modalities in the merged CCA modality. So each dot here is a sample. And I'm showing that sample's location along PC1 in the CCA or in the CCA representation of the data on the x-axis against that sample's location in either the mRNA shown in blue or the protein shown in orange representation. And basically what this is showing is PC1 is like the same in all of three of these cases. And just sort of like we shouldn't take this for granted, again, when we did the naive PCA of the kind of merged genes by cells matrix, so I guess it's not genes in this case, features by cells matrix for each modality, you really don't see that kind of correspondence. So what that means to us is that NAMPCs are kind of independently picking up on this same axis of intersample variation from each of these modalities. And that both reinforces our belief in the importance of the spectrum and also maybe suggests that CNA is managing to squeeze a lot of information out of each modality on its own. Whereas in contrast, this naive PCA isn't able to do this, maybe because there's variation across cells within each sample that's specific to each modality.
Yakir, there's a question in the chat, just real quick, by Neva Durand, who's asking, do you think you could apply this across different experiments performed by different labs, such as data sets from different papers or even ENCODE or other repositories?
So that's a great question. And I think it gets at some of the questions of sort of like in what's next vein. I will say here, we've only ever, right, and what I'm showing here, we've got different modalities, but the same cells. And that's really key. And presumably the same batch effects, right, because these samples were all run, this data was all generated in kind of one go. Something that we're thinking about these days is kind of like how, can you do sort of CNA type things on different data sets and combine them? And it's definitely a kettle of fish. I mean, the way that it is, I think, for all type of single cell methods. And I think there's going to be a lot of the same trade-offs that we have to make. Which is sort of a non-answer, but I think that's kind of where we're at.
Yeah, I might add one more thought to that, which is if you have good data set integration methods and you can combine those, multiple data sets upstream, then you could apply CNA to that joint data set. One thing that is important here, though, is that our neighborhoods are constructed and those result in our features within a given data set. So we don't have a natural way to, if we ran CNA in one data set, to then sort of project a new separate data set onto it.
Exactly. Dylan.
I just want to check that I'm following along with this graph. So basically what you're saying is you constructed neighborhood matrices based on the ADT, the protein data, based on the RNA data, or based on the CCA.
Yeah.
And then you find the PCs of those three matrices, and they agree.
PC1 agrees.
PC1 agrees. So you're getting the same PC1, whereas like PC1 from RNA data, PC1 from protein data, like generally don't agree nearly as well.
Yeah.
Okay.
Yeah, that's right. Okay, so this is PC1. We obviously also tried to sort of look at other PCs to look for, any sort of interesting things that they might correspond to. And one example that we think is really neat is that in the mRNA data, NAMPC2 and NAMPC4 seem to correspond pretty neatly to sex. So here in this plot, each dot is again a sample. They're colored by the binary sex reported in the data set. And you can see that there's a pretty clear delineation here. And when we looked into what NAMPC2 was, we found that it seemed to largely capture known abundance differences of CD4 and CD8 T cells between males and females. So there's this well-known thing that males tend to have a higher CD8 to CD4 ratio, which is what we're seeing here. NAMPC4 was a bit more surprising. So you can see here that the neighborhoods don't segregate neatly by color in UMAP space. They're really interspersed, the red and the blue. And when we looked into the genes driving membership in this NAMPC, we actually saw why. They're all genes that are expressed on sex chromosomes with the appropriate direction of effect. So exist is the most strongly negatively correlated gene. And then all these other genes, you see there's Ys in all of their names because they live on the Y chromosome, are all sort of driving the positive loadings. So this NAMPC actually seems to pick up on microscale structure in the nearest neighbor graph that's driven by expression of these genes. It's like there are adjacent neighborhoods of globally very similar cells that are just differentiated by their expression of these sex chromosome genes. And we thought this was a neat demonstration of the power of NAMPCs to pick up unanticipated structure, and in particular to pick up structure that you really wouldn't find in any sort of cluster-based analysis. And I do want to say that this result was conducted, this result that I'm showing you here is from data, it's from an analysis in which we removed the harmony preprocessing step in this data set that's intended to kind of smooth out batch effects. When we re-add harmony, this result is strongly attenuated, and it kind of makes sense, I think, why, because harmony in a sense is performing local averaging in this graph. And so it sort of probably kind of zeroes out this particular signal. So I want to turn now to a more supervised kind of mindset, looking at the phenotypes that we had in this data set. The most interesting, or the primary one of which was this phenotype of TB progression, whether people infected with TB are able to control the infection versus not. And the original analysis done by the folks who put together this data set was based on a very detailed clustering shown here. So this is 31 different memory T cell subsets. And this analysis found two clusters that I've shown at the bottom here, 12 and 20, that are kind of helper T cell clusters that were significantly depleted in patients who progressed to active TB. And this was a really big deal because it's the first time that a signal of that sort of specificity and fidelity has been found in an unbiased case-controlled design for TB progression. And for a disease like this that really devastates large parts of the world, it's really critical to understand why some people can control the pathogen and others can't. When we analyzed the same data with CNA shown at the right here, we found two things. The first was a broad recapitulation of this kind of depletion signal that you're seeing here. That's this blue swath of T helper cells. And then the second is actually this new result, which is an expansion of a cytotoxic CD4 population among TB progressors. And this was exciting to us because, first of all, it suggests that our kind of data-driven approach that we've baked into CNA does maybe lead to more power for association testing in practice. And it's also interesting because this data set was really the result of a very significant collection effort that spanned many person hours and years to put together. And so the suggestion of a second associated population here could open up new avenues for understanding the interplay between host T cells and TB. I'll say quickly that we were able to also find a significant association. What I'm showing you here is the CCA-based analysis, which is what the authors of the data set did in their primary analysis. In the original cluster-based analysis, the mRNA-only analysis was actually null. It didn't find anything. And one thing that we thought was cool is that actually CNA does find a similar result in just the mRNA alone. So again, kind of getting at potentially a suggestion of increased power here for this type of approach. Okay, so let me now move to our last analysis that I'm going to show. As I mentioned, in this data set, we had information not only about TB progression, but also about basic patient demographics like age, season of blood draw, and actually also things like that were genetically estimated, like % European ancestry, because there's genotype data for these samples too. And of course, we have to note this % European ancestry variable is likely just tagging significant environmental variation that co-varies with ancestry in this cohort. But it's there for us. And given this wealth of kind of information about the samples, we decided to perform a scan with CNA just to search for any associations to, I think, approximately 20 phenotypes that we had available. And these are some of our favorite examples of what we found. And what we like about them is that they include a mixture of known but also new associations. So for example, in this part of the AGU map, this red part lines up with a known positive association between CD8 T cell abundance and age. But this more complex blue area here among the T helper cells, and also this transcriptionally similar but oppositely associated area close by, seem kind of interesting for further analysis. Similarly, these two regions, the red and the blue here, in our results for season of blood draw, correspond to known seasonality of Th1 and Th2 cell abundance. These are T helper subsets. Though interestingly, actually in this Peruvian cohort, the directions of correlation to season are reversed compared to analyses in other parts of the world. But again, there's also these broad swaths of transcriptional space that don't seem very clustery and kind of demand further exploration. So the cool thing to us about this is that, you know, where we are in single cell today is that these are really fundamental questions, like what is the relationship between age and seasonality to our immune systems? And we're now getting to answer them at unprecedented resolution. And our hope is that CNA can basically take us one step closer to realizing that promise. So with that, I'd like to thank all of our co-authors shown here, listed by name and circled in red, and of course the Raychaudhuri Lab more broadly. I would also like to thank Shomo, obviously, for his guidance and support, as well as the other PIs who were involved in this project. And you guys for listening.
Thank you.